Welcome to the Wednesday afternoon lecture series. This is the final lecture for the season. I'm Dr. Francis Collins, or I play him on TV. Dr. Collins unfortunately could not be here today, but I'm Jim Cimino. I'm the Chief of the Laboratory for Informatics Development. It was partially the reason for inviting our guest speaker today, a friend of mine from way back. And I'm going to actually read the intro because I can't memorize all these titles and honors and stuff. So it's Dr. Butte, who's the chief of the Division of Systems Medicine at Stanford University School of Medicine and the Lucile Packard Children's Hospital. He's an associate professor of pediatrics, computer science, and immunology and rheumatology. His background is he has a Bachelor of Computer Science and an MD from Brown University, did residency in pediatrics and fellowship in pediatric endocrinology at Children's Hospital in Boston, then went to MIT for a master's in medical informatics and a PhD in medical engineering and medical physics. Since the 1990s, he's been developing knowledge discovery methods in clinical laboratory databases using techniques like relevance networks and data mining, and lately has been having some remarkable success using publicly available data resources to discover novel genome-phenome relationships, and I think we're going to hear about some of that today. He's got a number of honors. I think one of the best ones is that back in April he did a TED-Med lecture on, which is now up on YouTube, and that's maybe the short version of this, so you're going to get the longer version. And he's also been on the board of directors of the American Medical Informatics Association. He's a fellow of the American College of Medical Informatics. And in 2008, he ran the first AMI, was it Translational Bioinformatics conference in San Francisco, which he did the first one, and it's really taken off and become a major annual meeting in the field of translational bioinformatics. So with that, I'd like to welcome Butte.
Excellent. Thanks for having me here, and thanks for putting up with the heat, and actually for those of you here live for actually making the trek over to Building 10. I also want to just make one other point about introducing myself to the high school kids and the college kids here in the audience. I was in your seat back in 1991 as a summer student here. I spent the summer here. That became a year when I was in medical school during the HHMI, living on the Cloister. And then I got a institutional NRSA, then a T grant, then a K, then an R1, then U's and P's. And everything started with that summer here. And so it's a real honor to be able to give one of these lectures, since I benefited so much from listening to them at one point in my life. Now, the usual kind of disclosures: I've started a bunch of companies, I consult for a bunch of companies. You shouldn't believe another word I'm about to say here. So if you haven't heard, we're in the middle of a data deluge, according to The Economist magazine. This came out about 2 summers ago. The human species now generates 2 zettabytes per year. If you don't know your metric prefixes, it's kilo, mega, giga, tera, peta, exabyte, zettabyte. Next year, of course, we will generate 4 zettabytes because it's obviously exponentially growing. And in fact, we generate so much data now that my favorite article on this comes from Chris Anderson at Wired Magazine, who's the editor-in-chief. 2007, he says, we're generating so much data, science itself is starting to become obsolete. Okay? Now, let's be really clear. What on earth does he mean by that? Because you think about the scientific method for a second. The scientific method, going back, let's say 400 years, the scientific method is we ask an interesting question or generate a hypothesis, and then we go get and make measurements to answer that question or address that hypothesis. And now what happens in the world when we have so much data already? We have the measurements, right, in many fields: physics, astronomy, certainly life science in a minute. We have so much data that the new magic is in figuring out what's the cool question I want to ask of this data. And that is really 99% of the work in my lab now. What's the cool question we want to ask? You know, the whole world has been waiting for the answer for this one. And nobody even realizes this question is askable today because we already have the measurements. So in that way, the scientific method is kind of becoming backwards, where we're getting the data already. What do we want to ask of it? And it's certainly happening in life science research as well. The article at the top here comes from the Harvard Business Review. And it's a business journal, as you can imagine. It's great for junior faculty to read this. It talks about that's a great magazine to read. It tells you how to organize your lab and things. In 2010, they said data-driven science is the next big scientific revolution. You think about how we started with experimentation in the 1400s. We went to theory in the 1700s. We went to simulation and models in the 1900s. and now data-driven science in the 2000s and beyond. Nature says it is shameful if we don't make use of the data we put out there. Science says we need to make data maximally available. And the Lancet says if we don't share and use that research data, it's actually an impediment to public health. Now, where's all this data coming from? It comes from devices like this. Of course, I got to bring my props. On the left there is a standard Affymetrix gene chip. This is what it actually looks like in real life. It's kind of small. It's the size of your thumbnail. Here's the chip. Here's a thumbnail at the bottom. And as you all know, this lets us quantitate every gene in the genome, okay, at the RNA level, the blueprints from DNA on their way coding to proteins. And you all know, especially any of you who spent any time at the bench, we love to scale. When we get one of them to work, we love to make 96 wells of them, right? So this is the 96 well version of that. You can see one version of it there. Each well there has one of those microarrays at the bottom there. And you know, it's kind of amazing. With a chip like that, we can quantitate every gene in the genome. And it's really amazing that actually this is a 15-year-old device now. We've had this for 15 years. And in fact, it's a commodity item, right? I don't know about you guys. For us at Stanford, it's really, which is the cheapest vendor today, okay? And they don't like us saying it, but we really buy these things on price, right? $200, $300 gets us the chip on the left there, right? What's absolutely still amazing to me is that actually we make scientists share this data on the internet, right? And you know, it's not perfect, and not everyone shares right, and not everyone gives away everything they're supposed to give away, but it's amazing how much data we have on the internet just from this one high throughput measurement modality today. So one place you can go to get this kind of data is run at the NCBI, of course. A lot of friends, and my research career would not exist if not for the NCBI. Okay, for all you in the audience, I'm just putting a shout out for you. NCBI Geo, the Gene Expression Omnibus, is the repository for these microarrays. And you can see as of a couple weeks ago, we're up to 752,000 publicly available microarrays at the NCBI Geo. That's just the US repository. The Europeans run one called the European Bioinformatics Institute Array Express, run by EBI. And if you don't look at the overlap, you just count the unique ones there. Another 213,000 more arrays. Suffice to say, these are growing like crazy. We're just a few weeks away from having 1 million publicly available microarrays. That's up from 0 in 2002, okay? And the data continues to grow exponentially. I think it's doubling every 2 years now. You know, we're just finally slowing down to Moore's Law. You know, Moore's Law, computational power doubles every 18 months. Yeah, I think this is only just doubling at that rate now because it was tripling or quadrupling earlier. And it's only slowing down because of the next repository I'm going to show you, and you guys can guess which one that one's going to be. And here's just the most amazing point of this. And this is obviously directed to certain junior folks in the audience here. In the middle of this website, just in the middle there, is a very simple search box. And that search box, right here in the middle here, I'll get the brighter pointer here. A high school kid today who needs to do a science fair project, right, for high school. She can type breast cancer here, click go, and find and download 31,000 samples of breast cancer. About as easily as she can find a song on iTunes today. 31,000 samples of breast cancer digitally available. You know, if you look, that's more than 1,000 independent experiments on breast cancer. And what kind of question can they ask? Well, there are breast cancer researchers who are just absolutely religious about their use of human cancers and biopsies. And you got folks who say, well, it's science because I'm doing this on cell lines. You got these folks who are working on mouse models. And even a high school kid can just say, well, what's in common across all 1,000 experiments? I don't care if it was from a biopsy, a cell line, from a mouse, what's the common denominator, the core common denominator in breast cancer today? And you know the other thing about 30,000 samples of breast cancer, if you haven't figured it out, that's more samples of breast cancer than any breast cancer researcher will ever have in their lab. Because by definition, if they want to publish in a good journal, that one investigator has to put their data out, plus their competitors. So folks who get to the repository now and know what to do with this are going to have more samples than anyone else in the field. And if it's not breast cancer, it's colon cancer, prostate cancer, hundreds of diseases. If you're still kind of skeptical about it, I'm very proud of these 5 high school kids who either placed in Intel, Westinghouse, or Siemens semi-finalist or finalist pools from my lab in the past 3 years. Each of these kids was in the top 300 science kids in the country. A high school kid today, they get to meet Obama, they have an asteroid named after them, all that kind of stuff. A high school kid today has more digital access to samples than any researcher in the field today, if we choose to empower them with the questions they should be asking of that data. Now, if RNA is not your thing, obviously DNA repositories are growing like crazy. dbGaP is growing faster than Geo in some ways. And here, it's not so easy. A high school kid can't just click to download the data. We are a little bit more finicky about privacy and security with DNA sequences and genotypes. So here you click to download the permission forms to go get IRB approval. So it's a little bit of a barrier there. And here the stick is a lot harder. It's not the journals with the stick on the investigators, it's NIH and Wellcome Trust that make scientists share this data even before publication. So we got to tolerate with these embargo dates. We got to give them a fair shot at publishing this data before we do. Why is it worth all that hassle? I just want to show the second one here, which you cannot read, is the entire Framingham Heart Study, okay? The reason why we know the word cholesterol in the United States. Now, I think it's kind of nifty that with the permissions, I can go download 14,000 people's genotypes. But I think it's even more amazing. I could just download 10, 20 years of phenotypes and clinical measurements and research measurements. And those guys at Framingham, they're pretty productive and they're pretty nifty with this data. But if you think they've found every possible predictor for disease, you know you're wrong. Sitting in there might be the next big diagnostic for a disease that they just haven't thought about looking at yet. And in the old days, not so long ago, you used to essentially have to work there to get to this data. And now we can all access this because of data transparency, reproducibility. So there's rules here, but it's an amazing world. And just think for a second, that's just number 2 on a list of hundreds of such experiments here, waiting for you to ask an interesting question. Now, we've had this data, right? It goes back to GenBank 29 years ago, right? The idea that we should share research data. And we can look at this data and we can start to get ideas. This disease could be treatable with this drug, and we've done a lot of this kind of repositioning work, and this diagnostic could predict this particular disease. And where we used to get stuck, though, is how do we convince, as a computational researcher, how do I convince the biologist or clinician scientist how to test my idea, right? I'll give you an example. The other day, we had a great prediction, a computation prediction for a serum marker for acute myeloid leukemia, AML. That's a particularly nasty form of leukemia. You might want to detect who's recurring from that sooner than otherwise because it's a pretty lethal disease. You think about bone marrow transplantation. So we have a serum marker for AML that we think we want to validate. So we go to our cancer center. It's NCI designated. I love my cancer center folks. I'm a member of the cancer center. And I say, do you have any saved serum or plasma on AML? Okay, we have a biobank there. Do we have any saved samples there? And they said, you know what, microarrays were invented at Stanford. We've saved all those tumor cells and we've extracted the RNA and we can get any of the RNA on the tumors, but they never saved any of the liquid around the cells, the serum or the plasma. That's okay. They said, go get IRB approval, put up those posters you see in the clinical centers like yours and ours. In about a year, you should have enough samples to test your computational prediction. And what does an informatician like me do? We just Google for it and find a company like this. Conversant Bio, browse by disease. What are we studying today? Bladder cancer, brain cancer, breast cancer. Here's leukemia. Click on that. What do I want to buy? Bone marrow samples, CD19 positive cells, CD45 positive cells, mononuclear cells, T cells from bone marrow, peripheral blood B cells. Let's go to the next page. Okay, here's plasma from peripheral blood leukemia. Each patient, I got a de-identified ID, age, race, sex, alcohol, tobacco, all the drugs, all the samples available for this patient. $55 each. So we buy them all. And we validate our markers this way. It's kind of not a big deal to get cancer serum, plasma, pathology samples, tissue microarrays. Where does this come from? These are big hospitals in the middle of the country, not really affiliated with a medical school. This is stuff on its way to the trash. And they strip off a label, put on a new label, get some de-identified identifiers, And some interesting companies resell this to researchers today. This comes on 48 hours on dry ice. I can't beat this price. I can't even beat this kind of turnaround time with my own institution today. And so I love biobanks and biorepositories, but they better, at least the academic ones, better be at least as efficient, if not better, than what we get from a company on a website today. But then the purists say, right? The biologists say, well, this is all descriptive research, right? Who cares about this? I don't believe it unless you show it in a mouse model. It's got to be necessary and sufficient, right, for some process. And then I show them assaydepot.com. Kind of sounds like Home Depot, right? Assaydepot.com. Okay, well, do I want to run biology, chemistry, drug metabolism, pharmacokinetics, toxicology? Let's click on pharmacology here. Now, what kind of mouse do I want to run today? Bone, cardiovascular, dermatology, diabetes, genitourinary, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. Well, you're going to hear a little bit of a diabetes story from me, so let's click on diabetes here. Here's a standard OB diabetes mouse model. This is a 16 mouse experiment. This mouse has been eating a lot and getting diabetes since the 1970s. Divide up these 16 mice however you want. 2 groups of 8, 4 groups of 4. Test any drug you want. They do insulin tolerance tests, glucose tolerance tests, fasting blood sugars. Try any drug you want. This study is on. What am I covering up? The price. $9,000 for this service, 9-week turnaround time, and literally add to shopping carts. Now, entire translational mouse experiment is just purchasable with a credit card today, okay? Purchasable with a credit card. So, look, I'm not trying to disrespect anyone who does mouse studies, but if this is all you do, there's a company that's going to do it for you. Nobody pours their own sequencing gels anymore, remember? Right? So this is the logical extension from that, right? Now, some of you with very sharp eyes are sitting in front and say, well, what do you know? This is run in China, right? And you're outsourcing this, but maybe you're offshoring this as well. And is that really cool? Well, actually, when you click on Add to Shopping Cart, all 133 labs and companies who want to do this experiment for you compete for your business, including those in Maryland and Wisconsin. In fact, it's easier to order from this than Amazon. Well, let's say I just want FDA-approved ones, or ISO 9001, or GMP, or AAA-LAC, or USDA. Let's say I have to have GMP. Well, that's 20 of the 133. Whatever certifications you want, this experiment gets done. Now, I know some of you are dying to criticize me on the quality of the output from these companies. I'll just simply answer. If it's this cheap, run 2 of them from 2 companies. And this is what we do, okay? How many biologists go across the hall to get their neighbor to do the same experiment? They don't do that. We'll just order 2 of them. And so this is a world where the validation methods are commoditized. This isn't kind of rocket science anymore. People just sell these services today, even in the United States. So what I'm going to argue is that if you think about the translational pipeline, right? We've been talking a lot about translational pipelines as a community with NCATs and the others. If you think about we make clinical and molecular measurements increasingly high throughput, we ask interesting translational questions or trials, we run a trial, we apply statistical computational methods to that data, and then we validate a drug or a biomarker. Actually, I'm going to argue 3 out of the 4 today are commoditized. We have plenty of data. My goodness, the statistical and computational methods in my field, we put out so many websites and tools, it's hard to distinguish signal from noise there. And the validation side, that double check, that testing of that drug or the biomarker is completely commoditized today. What I'm going to argue, though, is nobody's ever going to outsource asking good questions. If there's one take-home point for the summer kids and for the college students and trainees, you're never going to go out of business if you know how to ask good questions. In fact, in my lab, we say outsource everything but the question now at this point. Asking good questions will never go out of style. Now, another way to think about it is this. I live in Silicon Valley now, and we're so used to even starting companies. Kids start companies like Facebook in a dorm room, Yahoo, Google starting dorm rooms, the companies starting garages like Apple and HP. I think we have so much going for us. I think the next Amgen, the next Genentech is going to start in a garage. Where are my garage biotechs here? Maybe even the next Merck or Pfizer. I have 1 million arrays in my garage. I have every possible mouse model purchasable for my garage, right? All that's left is enough room and some good soda and candy to just get someone going and just turn this crank and get the ideas flowing here, right? That's all you need to fit in your garage today in some ways. And so this is what we practice. We come up with findings, we spin out companies out of my lab. I just want to practice what I preach in this way. So I'm going to spend most of my time talking about a new finding. It just came out about 2 weeks ago in PNAS. And in some ways, with no disrespect to any of the other investigators in my lab and any of the other papers we've published over the past 10 years, 15 years, this is my favorite paper of them all, okay? And many of you might not have seen it, so I want to explain this paper and how we got to this point. And it addresses a particularly vicious disease called type 2 diabetes. It is the public health menace of the world. About 1/3 of all kids born in the year 2000 in the United States will have type 2 diabetes, knocking 1 to 3 decades off their lifespan. And it's particularly vicious, 12% of American healthcare dollars, 20 million in the US, 170 million people in the worldwide, and it's a world health problem. And we have a lot of drugs for diabetes. We still need new drugs for diabetes. The latest ones, for example, DPP-4 inhibitors came out in 2008, which affect glucagon signaling or secretion. We can affect how insulin is released. We can affect how insulin has its effects. And these drugs just have immediate uptake, reaching billion-dollar markets because we just still need new therapies for diabetes. We still don't know really why people get type 2 diabetes. And nor do we really have a universal kind of biological mechanism there. So just to address one of the hardest conditions, we said let's just take our informatics approach and apply it to type 2 diabetes. And so a lot of it actually got inspired from this paper, Questioning Standardization in Science, from Nature Methods, April 2009. I just want to take a minute to explain what this paper showed. This paper comes from the field of mouse psychiatry. So think about what it means for a mouse to have anxiety and depression. Sometimes it's the only way to study the genetics of these kinds of psychiatric diseases. And these guys were kind of annoyed and depressed because none of their answers were matching each other. Because one group had one way of thinking about depression in a mouse, and another group said, well, actually, depression in a mouse means this, right? And how do you actually agree? So what they did is, 10 years prior, they said, well, we're all going to agree. We're all going to agree. This is what it means to have depression in a mouse, and anxiety in a mouse, and attention deficit in a mouse. We're going to agree what those standards mean. And lo and behold, all of their answers start to match. Everyone starts to get the same answers. But why this paper? Because all of a sudden, the minute they tweaked any one parameter in their experimental model, they changed what kind of water they used, or they changed what kind of chow it was, what strain it was, all the answers were not generalizable. The entire field essentially had overfit their model. They all agreed on this one model, they've all standardized on it, and they all get the same answers, and it has zero relevance for anything else today. And what this paper was saying is, actually, maybe today we should deliberately do different things, okay? Why standardize? Let's try different experimental conditions that are all logical. And if we end up with something in common, maybe that's the most resilient of the answers out there. And that is the causal mechanism. So let's take this to heart and study type 2 diabetes. And by studying type 2 diabetes, I'm going to look at micro-experiments. I already showed you there's almost 1 million of them publicly available, coming up soon. Almost 1 million. And so when I look for experiments, what I'm looking at are experiments that looked at samples from diabetes and samples from control. Diabetes and control, diabetes and control. But I'm looking at fat, muscle, liver, beta cells, the cells that make the insulin in pancreas, from rat, mouse, or human. 3 species, 4 tissues. I'm just simply asking what's in common. What's in common? So how do we do this? Well, let's go make a list of every gene in the genome and just simply count how many times does that gene show up as being different in one of the 130 experiments. So here's a graph that goes from 0, 130 would be up where the flags are. It ends about 75 here. And let's just start by making a list of every gene in the genome. Let's take 25,000 genes in the genome in the background. Almost every gene in the genome, 1/4 of the genes in the genome, change in expression level in one of the 130 experiments. It's not that rare to get some gene to change in some experiment. And there's a kind of quick long tail, kind of weird bulge around 20 or so. So a lot of experiments change in certain tissues more than others. But you can see there's a big curve to the left here. Most genes don't change in many experiments. Let's take some genes that we know have something to do with diabetes. The ones from these so-called GWASs, genome-wide association studies, like TCF7L2, We have a leptin receptor, which are not really GWAS hits, but other known genetic hits. We have some drug targets like PPAR gamma, insulin degrading enzyme. And all you can see is that the known genes seem to be changing in more of these microexperiments than the background list of every gene in the genome here. So we're staring at this, and we're staring at this, and we're staring at this, and we just simply ask, what's this little red dot over here? There's a gene changing in 78 out of the 130 experiments, 78 out of 130, and it's not one of the gold standard ones. No one's pursued it for type 2 diabetes yet. So I'm going to call that gene A for the moment, okay? I'll tell you all the answers at the end, but let's keep some suspense here. Gene A. And we're looking at gene A, and we're looking at gene A, and it's got a very interesting symbol, gene symbol. The symbol starts with CD. Okay, so that means it's a CD molecule. So that means it's a gene that codes for a cell surface protein that someone somewhere flow sorted cells on, right? So it's a cell surface protein. In fact, it's a functioning receptor. And we look a little bit lower on our list, and gene B, and this red dot here, is the ligand for that receptor. That's what binds that receptor. So now we're really interested because this receptor has never been pursued. So this ended up being a lot of collaborative work with many labs across Japan, especially the University of Tokyo, Keio University, Kitasato University, and Riken, all run by my postdoc, now staff member, Keiichi Kodama, and had a whole network of friends helping with this study here. Where we didn't have a collaborating lab, you know how we purchase these things, okay? Because we're not going to let that slow us down here, right? So we go and we look at our database, and the database says, well, if you're going to pick a tissue, go chase this down into adipose. Because the adipose, the fat, really seems to change this gene a lot, followed by islet cells, then liver and muscle. In fact, this graph says 90% of the adipose experiments where the receptor changed, the ligand also changed. So that means it might be what we call an autocurrent effect. The ligand is being made probably in the same place that the receptor has an effect as well. So now we can just get some wild-type mice, what are called C57 black 6 mice. You can see some graphs here. Give it a high-fat diet, the receptor level goes up. We start to make pictures of the fat. We stain for this CD molecule. And you can see, interestingly, it's not the fat cells, but it's these inflammatory cells around the fat cells. And we think they're macrophages, but there could be other types of inflammatory cells there as well. And so this is not a fat cell issue, but this is the inflammatory cell around it that's making this particular CD molecule. The graph here just shows the ligand and the receptor are indeed co-expressed. At least there's a correlation between them by qPCR at the RNA level. So we look at this receptor and think, boy, it's kind of interesting. It is actually in the fat tissue. And we start to think, well, maybe we should knock out this gene in a mouse. And then we go to Jackson Labs, and we realize this gene was knocked out in a mouse 11 years ago by the immunologist. Being a CD molecule, you can guess the immunologists were interested in this one. By the way, this receptor was cloned in 1985, okay? This is a low number of CD. It's not even triple digits here, okay? I'll tell you the answer at the end. So it's been knocked out 11 years ago. No immunologist did put a glucometer on the mouse. They didn't look at the sugar levels of the mouse. They didn't do a fasting study or any of that. So it's about a $6,000 experiment to go get this mouse from Jackson Labs. So we get the knockout mouse, stain for macrophages. You can even see from the back of the room, there are fewer inflammatory cells. It's not zero. Fewer inflammatory cells in that mouse that's missing the receptor. And over here, you can just quantitate that. But how does the mouse do functionally? The mouse without the receptor does better than the wild type. It's more insulin sensitive. So in other words, it doesn't die from diabetes missing this receptor. It does better than the wild type mouse. Whether you look at fasting blood sugar, glucose tolerance test, insulin tolerance test, normal fat, high fat diet, no change in weight, no matter which ones you look at, this mouse is insulin sensitive compared to the wild type here. So this tells us lower is better. If we can knock out this function, boy, maybe this is a new therapeutic target for type 2 diabetes. At least let's treat diabetes in the mouse, okay? We got mouse data so far. Let's connect it back to humans. You can buy slides of fat from human. There is plenty of fat to go around from liposuction and cosmetic surgery. Companies out there, they put these on slides, they sell them for a couple $100s. Here's a 36 BMI, a BMI 36, almost 37 BMI, 57-year-old woman. This fat comes from her. chock full of macrophages making this CD molecule, this receptor. So it's not just a mouse thing, human fat has it as well. And we look and we look and we see, you know, this CD molecule isn't just on a cell surface, there's a soluble form in the blood. You can just measure it in the blood, in the serum. Well, that's a $500 ELISA kit. And the level in the blood correlates with something called the hemoglobin A1C. That is the blood test we use in diabetics. Actually, that's what we follow once you're diagnosed with diabetes to see how your blood sugars are doing. Lower is better, lower is better. The less of the receptor in the blood, the less you lower your hemoglobin A1C is. By the way, these 55 folks didn't even have diabetes yet. So already, as this level is going up, your hemoglobin A1C is going up, or vice versa, but higher is worse, lower is better. It actually correlates with something called a HOMA-IR, which is a proxy for your measurement of insulin resistance. Everything is telling us lower is better, lower is better. This is a great therapeutic target. So he said, oh hell, let's just design a drug against this one. So how do you make a drug? Simplest possible drug. Here's the receptor. Let's just treat these mice with anti-receptor antibodies. We're going to treat wild-type mice with anti-receptor antibodies, and as a control, isotype control, other antibodies. So what happens to the mice? These are wild-type mice, high-fat diet. In about a week, we can wipe out the inflammatory response, not 100%. In about a week, you can wipe out the inflammatory response, and in about a week, we can lower their blood sugar. So let's just recap here for a second. We got a prototype new drug, we got a serum companion diagnostic, we got the knockout mouse, we got the human and mouse immunohistochemistry. Took about 18 months of work, and we did it with the same data any high school kid can get to today. And just sitting in that public data are many other findings like this. We've now since started doing this for type 1 diabetes, scleroderma, lupus. We've done it for 6 cancers, again and again. You know, the kids call this crowdsourcing, right? You know, when you get your whole friend community. This is retroactive crowdsourcing. We've had great scientists generating this data. Now let's pretend we told them to do this, right? And use the data forward thinking. The data is sitting there. It is high value. For some reason, we think if it's free and on the internet, it must be valueless, right? Because we've seen plenty of cats playing piano videos, right? I'm telling you, in NCBI, free and on the internet is extremely valuable. Podunk University doesn't get grants to run microarrays. It's the best universities, the best investigators. It's our peers that generate this data. But for some reason, the minute it hits NCBI, we think it's valueless for some reason. I'm just telling you, this is what's possible with the data we put out there today. So this gene A is called CD44. It's the hyaluronic acid receptor. It binds many more things than hyaluronic acid. The ligand is called osteopontin, just to connect the dots here. And the osteopontin knockout itself was published in a JCI paper. It is actually insulin sensitive, so everything's in the same direction here. But osteopontin as a ligand binds something like 30, 40 different receptors. They never made the connection to this particular receptor. Anti-CD44, believe it or not, is in drug development today for, of all things. It happens to be a marker for cancer stem cells on top of everything else. Now, it might not be the same CD44. It's a complicated receptor, 20 exons, 10 alternatively spliced, radically different shapes with the alternative splicing. Who knows if the one in the cancer stem cell is the same one that's affecting the fat and maybe the liver and beta cells. But there are people trying to develop this as a therapeutic for the cancer field, and maybe we should just go peek and see what's happening to the blood sugars on these patients as well. So there's a lot of future, I think, for this molecule and many others that we're finding this way. Now, obviously, you know that diabetes isn't just going to be a problem of genes and gene expression. Why are we having an obesity and diabetes epidemic? It's really because of our environment, right? And you can blame it on a lot of things. You can blame it on Cheesecake Factory-sized portions, 1,000 channels of TV with a sofa in front of them. Kids don't have recess in high school today. You know, standardized testing and all the negative consequences. There are a lot of reasons for diet and obesity leading to the diabetes epidemic today. But are we really so sure there's nothing else in the environment that's leading to the diabetes epidemic? And so the people who study the environment are the epidemiologists. And I have great friends and colleagues who are epidemiologists. But the epidemiologists today, they have a great field. They look at one environmental factor. And if they show it's positive, you show it a big cohort, they get a JAMA paper out of that, or New England Journal of Medicine paper. And the last time the geneticists looked at one gene to start was like 15 years ago. The geneticists start by saying, let's just look at everything. Phase 1, get some hits. Phase 2, validate, and subsequent papers for deep sequencing. They start with looking at everything. So we said, why don't the epidemiologists, environmental scientists, think about just looking at everything? Because everything in the environment is huge. It's unbounded, right? It's not just, 6 billion base pairs. It's a huge number of things that could be in the environment. People have started to think about the exposome and envirome. These are not my words. People don't like people making new ohm words. But these were in the Science article that came out from Martin Smith and Steve Rappaport. But the idea here is, why can't we start to think about environmental causes the same way we think about genetics causes? Instead of going after one by one, start with as many as we're measuring, and then try to associate them with a particular condition. And so we did this as a prototype. We started this experiment. Chirag Patel was a grad student, now postdoc in my lab doing this. And how to do this? Well, we borrowed from another publicly available repository known as NHANES, the National Health and Nutritional Examination Survey, put out by the CDC. This is a function of the CDC. It's not research. This is how they inform Congress as to the health of the United States. In fact, when you are born, whether you were born in the United States or your children were born, undoubtedly their height and weights were plotted on a growth chart. Those growth charts come from the CDC cross-sectional data from NHANES. Very few people realize, though, that the CDC has been measuring hundreds, if not thousands, of pesticides, heavy metals, toxins on all of these Americans. These are randomly selected cohorts every 2 years, and they look at blood and urine tests on hundreds of thousands of individuals from those cohorts. And can you believe it? All that data is publicly available for you to do any science you want on this. So we said, let's just look at their fasting blood sugars and classify them as having diabetes or not, and look at all of these 4 cohorts, 1999 to 2000, 2001, 2, 3, 4, 5. And the way to read this, which you can't read, is in this cohort they had 13 polychlorinated biphenyls, so PCBs measured. These guys had 16 heavy metals. These guys had phthalates and phenols and even viruses, volatile compounds. Each cohort had a different set. And to be clear, these aren't static numbers, because sometimes it takes the CDC 5 years to actually finally measure the measurement that they made from the cohort 5 years ago. So these numbers are constantly changing. And so we said, let's just run every single one of these in a logistic regression equation, control for age, sex, socioeconomic status, ethnicity, BMI, and see, does any one of these factors associate with that fasting blood sugar for type 2 diabetes? This looks like 200 boring logistic regression equations. The reason why it was written up in 100 newspapers is because we called it an EWAS, Environment-Wide Association Study. And we plotted it on a Manhattan plot, which you're used to seeing chromosome by chromosome. Now we'll put the nutrients here, phthalates here, and we have a Bonferroni line in red here. And we're going to use nature genetics rules. We're only going to talk about the ones that are positive in 2 cohorts or more. And lo and behold, heptachloropoxide, the derivative of the heptachlor pesticide that was banned in the 1980s for cancer risk, heptachloropoxide is elevated in the blood and urine of Americans with higher fasting blood sugar at odds ratios of 1.8 and even 3.2. And by the way, if it hasn't hit you, those numbers are higher than any SNP or gene in the genome for type 2 diabetes today. Now, we've done this for lipids. We've done this for blood pressure. You're going to see a lot of these kinds of papers coming out, but I think this is the way we should start thinking about the environment. Some enterprising folks in Berkeley now have started to come up with enviro chips. Let's just measure 1,000 of these environmental factors. Some of you who are really early adopters want to purchase something called a Fitbit. It's a little thing that sticks in your pocket that counts how many times you're walking up and down stairs and how much exercise you're getting. You're going to be able to get measurements on people, a lot more people, to sample that environment. And we shouldn't be ignoring that environment. I'm going to bring it to the end, at the end, just bring the whole story together. I don't want to miss the environmental factors anymore either. So now let's put it together, right? You know we're in this era of sequencing excitement. What we used to call next generation sequencing is now previous generation sequencing. And we're rapidly heading toward an era where we're going to get whole genome sequences on a lot of our patients, if not all of them. I'll talk a little bit about Helicos in a second with their $30,000 genome. I think Helicos is already delisted on NASDAQ. There are many others coming and going. PacBio is on track to sequencing an entire human genome in 15 minutes, prompting Discover magazine 2 years ago to come up with the article titled The Jiffy Lube of the Genome. Those of you who drive a car know that Jiffy Lube promises to change your oil in 30 minutes, and this will eventually be faster, if not cheaper, than that. They generate, by the way, Eric Schatt tells me, they generate 20 terabytes of data every 15 minutes, night and day at PacBio today. So you can just get an estimate of the size of the data. We have two companies that are already offering, in quotes, $1,000 genomes, Autocurrent, $1,000 plus labor and reagents. Illumina is close to that as well. I think the current pricing, at least academic pricing, is something like if we order more than 100 genomes, our price today is about $3,000. What an amazing sentence that is, that people order these in units of more than 100, okay? That's just, when you think about it, it's kind of stunning. Complete Genomics is on track to sequencing 80 genomes a day if they don't lay everyone off. Cliff Reed says back in September 2011, we're on track with just linear trends to reach $33 per genome at the end of this decade, okay? Some of you might pay more than that just to park around here in Bethesda. So suffice it to say, it's going to be $0. I'm just going to say it's going to be negative dollars. There'll be companies that think it's worth for them to get your genome if you're willing to suffer some discounts. I think there's car insurance companies that already give you a discount on your car insurance if they place a GPS in your car to see if you're a good driver. Why wouldn't this be any different? You can imagine all sorts of scenarios. It's essentially be zero, if not negative, let's say in the next few years. I'm not even going to show you the Moore's law graph on this one that everyone always shows. And by the way, so one kind of quick take-home point, this is not theoretical. If you want to start playing with this data, just go to Complete Genomics website and just download 69 whole human genomes. And that way it's not theoretical for you. In fact, I was joking about this earlier. Problem set 4 in my class now that I teach every spring. Problem set 4 is go download all 69 human genomes, unpack them all, cluster them, and the right answer better be you get back the migration of people on planet Earth. So Africans here, Middle East, and Asians, then Caucasians. Well, I should be able to see that migration when you cluster this data. But by the way, you have only 1 week to do this because your final projects are due the following week. I can expect now undergrads to just cluster, download, and deal with 69 human genomes. And they all do it. And so we shouldn't be scared of this because this is what the next generation is going to be doing, and we can either get with the program or just be scared of this. Go, if you have any interest in what this stuff is, you can click it out. You don't even, I think you need to sign up with a permission form, but it's just free, clear data. Just start to play with it. So we were working with Steve Quake, and Steve Quake, I'm sure, has given talks here, and you know Steve Quake is a very entrepreneurial kind of guy. He published in Nature Biotech. He was like one of the first faculty members or something to get his genome done in 2009. And he started a company, Helicos, paid $30,000 for his company to get his genome sequenced. He released the data in yet another NCBI repository, the Short Read Archive. And so this was a landmark paper in 2009, Nature Biotech, but it wasn't clear what's the medical relevance of having your genome sequenced. And so what happened was Steve Quake ended up presenting as a patient. And so let me walk you through this. This is a paper that we wrote on him presenting as a patient. Ewan Ashley is a cardiac geneticist with his team, me and my team. Russ Altman, pharmacogenomics expert, who's given a talk earlier in this series this year, and his team. Steve Quake, of course, Hank Grilly on ethics, and George Church is essentially on every paper in this field. And so I'm a doctor, so let me present a case to you. Here's a 40-year-old male presenting to his doctor in good health, no real complaints or symptoms. Why is he presenting this family history of sudden death? Here's Steve Quake, and here is his nephew that died at 19 years of age, didn't wake up one morning, sudden death, sudden cardiac death. So Steve Quake now presents to his cardiac geneticist, Ewan Ashley, and says, Doc, am I at risk for sudden death too? Here's my genome. Okay? Patient has a heart rate, blood pressure, blah, blah, blah. Patient presents with 2.8 million SNPs, 752 copy number variants, and by the way, doctor, your next patient's already waiting for you in the waiting room. No one's ever going to give us more than 15 minutes to deal with this. So this paper became a prototype exercise of what it could be like in the future for a doctor to deal with a patient with a genome in a 15-minute encounter here today. That's what, look, we did some things right, we did a lot of things wrong, but it just became a prototype in that way. So first, the cardiac risk. Ewan Ashley could give an hour-long talk on this. I'll show one slide from him. Suffice it to say, it was not an easy answer. There are many genes that are known to have variants associated with sudden death. Well, oh good, lucky. Steve Quake doesn't have any of those known variants in those known genes. But he's got other new variants in those same genes. what are called private. They're private variants because we haven't sequenced enough people on planet Earth yet to know how many people have this. So we can run all the computational predictions, SIFT, Polyfin, 20 others out there. They all give different answers. And in the end, what could you tell Steve Quake? Well, Steve, instead of extreme exercise, why don't you limit yourself to moderate exercise? If that doesn't sound wishy-washy to you, it's definitely wishy-washy to me. The other problem was coronary artery disease. The other way you can kind of die suddenly is with a heart attack. And there is a history of coronary artery disease in his family. He's got a variant in this gene called LPA, which sets him up to have coronary artery disease. If you plug in his age and lipid levels, his phenotype into the Framingham risk calculator, the risk score would say not to start on statins. Based on the LPA, we said we should start you on statins, Steve. It's been written about in many newspapers. He's decided not to start on the statins. We have yet to find the gene in the genome for compliance with medicine. And he's undoubtedly got the risk allele here. This could be an hour-long talk on the cardiac. I'm just going to show you one slide. Another hour-long talk could be from Russ Altman. Indeed, I'm sure he's given that hour-long talk earlier in the sequence here. And the long story short is we could say something about 150 drugs today. Given a genome, 150 drugs. If he decides someday to start on statins, he's not going to get that kind of lethal myopathy. Warfarin, my God, we could say so much about warfarin, fluostatin. If you might respond to this one, fluostatin may be better than pravastatin. Suffice it to say, 150 drugs, we can say something about the dose, whether it might work, whether there might be adverse events. That could be an hour-long talk. Ross has given it. I'm not going to talk about it here. My lab was tasked with all the rest of medicine. And we said, this must be simple. There must be a master database somewhere with just all the disease SNPs, all the variants in the genome, and just how do we actually intersect that with someone's genome here. So these are the ones we know and love. The GWAS catalog is great. I talk about it every talk I give. When I looked at it a couple months ago, it had about 1,000 papers curated. People read this data. You got about 5,000 SNPs, about 5,000 different diseases. The problem is almost a third of the records there didn't really record which was the risk version of the gene and which was the protective version of the gene. Which allele was which, making this unusable for personalized medicine. Now, I love this database. We use it like crazy, and this is done with next to zero resources intramurally here. But this is the best we have. It's only the GWASs. When we have a positive hit and 50 people have validated it, those 50 don't show up in the GWAS catalog either, nor do any of the candidate gene sequencing that's been done prior to GWAS. NIH-GAD is another great database, 56,000 papers curated. So it's gene-based portal. You can type in a gene and see what diseases are associated with it, but it wasn't curated at the DB SNP level. So we cannot look at exactly which SNP was which. We just know the gene was involved. OMIM, everyone knows, it's wonderful. Mendelian disease, not the common stuff. HGMD is a commercial database, pseudo-academic. Many papers curated, many mutations, but even there, they didn't write down what was the odds ratio, which genotype was which. Is it the A or the T? Which was the worst? Which was the better? So we knew this day was coming in my lab. And so 4 years ago, we said we got to just start curating, rereading every paper in human disease genetics. Just got to read them all. Here's an example of a paper published in 2008, Journal Inflammatory Bowel Disease. Here's a great example of a post-GWAS paper. They look at Crohn's disease, ulcerative colitis, 9 loci, 700 finished patients. From this paper and others, we record more than 100 different features. Well, it was a GWAS, non-GWAS, what's the exact disease, like diabetes, phenotype, like fasting blood sugar, the population, the gender. We were talking about ethnicity. We have more than 800 terms for ethnicity in this database. Of course, we write down the alleles, the genotypes, the p-values, the confidence, the odds ratio, what technology, was it case control, longitudinal, and we map everything to something awesome called the Unified Medical Language System, the UMLS. Every one of you in this audience probably knows some piece of UMLS, whether it's gene ontology, ICD-9, 10, SNOMED, it's the HUPO, those gene symbols, and UMLS is now a 23-year-old initiative from the National Library of Medicine to put and intersect all of those terms together. So we map everything to UMLS. And how does this actually look like? A lot of it exists in annoying tables like this. RS1004819, C allele, T allele, this percent, that percent, and we have to multiply these all back to figure out how many people were in each bucket here. Here's another paper, 2009, a year later, Journal Rheumatology. They looked at ankylosing spondylitis, a different disease, 8 SNPs, this receptor, 2,000 UK case control patients. So this was from Wellcome Trust. And this table is sideways. You have to rotate it. RS1004819, here's the A allele. You can see all the percentages. I love computer science. I love natural language processing. You can start to see why this is hard. NLP has a hard time even figuring out where the tabs go here, right? And which allele was which. There's no standard way of looking at this. So by the way, some of you who have really sharp eyes noticed I showed the same SNP in 2 papers. And the first paper says the alleles are C and T, and this paper from the Wellcome Trust says it's an A. But most SNPs have one choice or the other choice. So how do you get an A here? What exactly are the alleles for this SNP? So we've got to look each one of these up in dbSNP. And in fact, the alleles are C and T. Wellcome Trust was wrong. And then we realized 11% of all papers in human disease genetics report on the probe they used and not the double-strand DNA of the patient. So now we've got to fix them all, and we have. And now you see why this is such a damn hard problem for computers to do. So we've done that. That's one paper. Here's 6, here's 100, here's 1,000. We've read more than 10,000 papers, 140,000 SNPs, 3,000 plus diseases, way over 500,000 records, and this is changing hourly for us. And we're just going to keep going like crazy with this. Published a lot of this, how we do this in this PLOS1 paper back in 2010. But now that we have this, we can start to make all sorts of interesting influences or inferences on the patient. So for example, Steve Quake, now we know he's a 40-year-old white male Californian. 9%, he starts with a 9% chance of getting Alzheimer's disease. Let's add back the SNPs. We're going to add them back with 6. We're going to believe the most believable ones are at the top, the least believable ones at the bottom. This SNP was in 6 papers, 5 papers, 4 papers, 3, 2, 1. And these are the number of people in each of those papers. And now if you're a conservative doctor or patient, you can kind of draw the line here and say, I only believe it if it's in 3 papers. My odds are now, Steve's odds are 3% instead of 9% of getting Alzheimer's. If I'm a young whippersnapper doc or patient, I believe everything in the genetics literature. I follow down to the bottom to this weak paper with only 170 people, and now I think my odds are 1%, my post-test probability. I'm not going to tell you which of these are right or wrong. You can decide with your confidence levels and your conservative nature or not here. If we can do this for one disease, we can do it for all of them. And so this is what we call a riskogram. Each arrowhead here represents the pre-test probability based on prevalence. By the way, it turns out just figuring out where these go is the hardest part now. We don't have any master list of how many Americans have every disease, right? We don't. We have for DOD, VA, some HMOs, we have for Medicare, Medicaid, but in general, we don't have a master list of how many people have which disease. So just getting the prevalence numbers is next to impossible. So we Google each one of these to figure out someone's best estimate. That's the pre-test probability, and the lines indicate the post-test probability. By the way, for those who are really aficionados in this field, because we think in pre-test and post-test probabilities and not odds ratios, that whole kind of scary incidentalome problem goes away. The incidentalome problem is that if I sequence enough genomes, I'm going to see random stuff that's going to make me spend a lot of healthcare dollars on you to prove that you don't have that kind of rare thing. And let me just make it really crystal clear. If I have a 1 in a million disease, 1 in a million chance you're going to get it, and now I tell you you're 100 times more likely to get it, it's still 1 in 10,000 chance you're going to get it, which is close to rounding error down here, compared to the stuff that's going to kill you someday at the top. If we're going to spend money on preventing anything, it's probably going to be the stuff at the top here. That's the point here. So what are we going to do about the risk, right? And that's the final point I'm going to talk about. I talked about the environment. I talked about pesticides, right? I think we can start to figure out what are the environmental influences that are influencing our risk of getting these diseases. But I think the environment is the new prescription for the doc, okay? In other words, what can I do to compensate for my genome? Yeah, I love gene therapy and all of that, and changing my DNA, and that's like still years away the last time I look. What can I do today to my environment to change or to influence or compensate for my genome? We came up with a figure like this, and the way to read this is that the same diseases now are on font size, so bigger font size means higher likelihood of getting the disease. Some diseases cause other diseases, so if you end up with these, you might end up with that. And all along the edge here are known published factors that influence the etiology of getting that disease. And we came up with a kind of trick to mine the hell out of those mesh terms in PubMed. Everyone knows about the mesh terms, but there are these sub-qualifiers, and some of them are etiology and adverse events. We mine those to figure out in an automated way how to draw this. So, Steve Quake, you shouldn't smoke, you should exercise, you should watch your diet, you shouldn't drink too much alcohol. Probably didn't need a genome for those 4. But Steve Quake, you had slightly higher risk for Parkinson's disease, and in 10 papers, pesticides, in tiny font here, influences the etiology of Parkinson's disease. This is crude. This is qualitative. This is first pass. But this is a way I think docs and patients can talk about their genome. You know, you're doing great on exercise. Let's work on your diet. Come back in 6 months. Let's see how you're doing there, and maybe we'll work on your alcohol, right? I think doctors and patients are supposed to talk this way with each other. So just in closing, to leave time for some questions here, let me skip forward a little bit here. Take-home points, molecular, clinical, even epidemiological data, and tools already exist. I think we can get to diagnostics, therapeutics, disease mechanisms. I think integration is an incredibly powerful mechanism. Why do I trust the microarray results from one lab when I can see what's in common across 100 of them? Personalized medicine is great when we think about DNA, but personalized medicine is greater than and equal to DNA. It includes that environment, includes clinical measurements, molecular measurements, besides just DNA. We shouldn't equate those two. For the bioinformaticians in the audience, I just have to encourage you to think that bioinformatics is more than just building tools. Think carefully whether the world needs yet another database-backed website to do something that people have already been doing. If you know your tool the best, I encourage you to use the tool. And don't just show people the URL, how to get your website, show the world what you found with your tool. I think you're going to get uptake and you're going to be able to change the world that way. The hardest part for me is this, is how do we find new investigators in this field that are willing to put a career, stake a career on publicly available data? Because unfortunately, when I see the biology grad students go to their first rotation, right? They're in grad school, they go into the lab, they're starting a rotation project, they pick up someone else's data, and they go and they're frustrated, and they're not getting the experiment to work. And what does the principal investigator in the lab say? If you didn't collect that data, you can't trust it. What a negative way to do science today, is all I can say. When we have thousands, tens of thousands of well-funded labs generating data on taxpayers' dollars, and the data is actually already publicly available. Again, it's not perfect. There are many people who are still not sharing, and they should be. But the data is out there, and I just encourage you to think about that data as being high-value data to launch on these experiments. I'm always looking for postdocs. We're also hiring faculty. I couldn't have done any of this without a long list of collaborations, especially a lot of different labs at University of Tokyo, Kitasano, Riken University, Keio University, and others. Obviously, Ewan Ashley, Mike Snyder, Steve Quake on the genome stuff with Ross Saltman, and many others. And I get generous endowment support from the Lucille Packard Foundation for Children's Health. My lab is blessed with 16 NIH grants from these 9 institutes of NIH. These 4 give me more. These 5 give me less, but I still love them. And just for any program officers in the audience, I still don't make that $1.5 million threshold that brings me in front of the Inquisition Committee. So a lot of this is good citizenship to participate in other people's grants here. Marcia Dines, Hewlett Packard, HHMI, stem cell propositions, which my state is particularly good at passing, internal cancer grants and stem cell and seed grants. I thank my admin and tech staff wherever I go. I'd never get a good grant or paper out the door without them. And I always thank my wife, who starts companies with me now, molecular biologist, biochemistry, reads every major paper and grant that comes out of my lab, and who lets me go all over the place to give talks like this. Thank you very much.
Thank you. We have time for questions.
Surely.
So you've told us before about some of the innovations you've made and some of the people you've worked with, the innovations they've made in text mining. You alluded to some. Can you speak a little bit more about that?
Absolutely.
Please? Thank you.
So I was kind of glib about kind of dismissing natural language processing, and we're not even strictly using NLP. It's more concept identification, even more crude tools. So how do you figure out what's in 1 million microarrays? You can't human eyeball those. That's more than these papers are. So we use concept identification to figure out what kind of data is in those. So for example, those postdocs have to write that annotation when they upload the data. So we use some concept identification like SNOMED, ICD-9, 10 to look for diseases there, and then we human eyeball them to figure that out. So we absolutely use the UML's terminology for the concept of identification. But some of these things are just, by the time we figure out how to get NLP to read the tables, we'll have already curated all the literature there. So at some point, you just have to say, I can't solve this computationally. I'm just going to hire a team to do this. Because we want to do the science with the results of that thing, right? Not just figure out how to get it to be done in a computational way. Let's go to you next.
You take publicly available data sets and publicly available information, and you create derivative data sets from that,
That's
like
right.
the 130 microarray things from there. Do you post those as publicly available data set for other people to download and to ask questions?
Yeah, so that's a great question. So questions on how much do we reissue the 130 data sets there? In general, we don't, because to be honest with you, there's no real place to put new annotations on that data. So to be strictly clear, an investigator might have said, well, here's an OB mouse and here's a wild-type mouse. But to us, we read that as diabetes and normal. So we have our own annotations on top of the annotations in Geo or Array Express. Right now, there's no place for people to just re-annotate that kind of data. I think folks, Elvis Brausman and others at EBI have been talking about building distributed annotations for data. But the raw data itself, in general, to help reproduce experiments, we give out some of the R code to do this. But we don't want to step on the original investigators' data. We want them to cite their data, really. So, but I don't know if I answered your question.
Not really.
Okay, let me try again.
The original microarray data sets were not in a sophisticated database. Joe DeRisi and Pat Brown, they posted them on their websites. They just made them available to other researchers, and I think that would be extremely valuable if, because it's really hard to get large data sets out of Geo and normalize them and all that.
Yeah, I agree. So I think you're right. So a lot of times, so for a lot of the papers, we put like the matrices on our lab wiki because there's no other good place, official place to put that kind of stuff. But I think what you're really getting at is that it is still hard to do. And I'll admit, I made this seem easier than it is, okay? Mapping the annotations of a chip from 6 years ago to today, these are hard problems. In general, what our methodology has been is let's at least share some of the tools we've used in our findings. So we give out the tool to map the arrays. It's called ILUN. We have a geo-based browser called Gene Chaser. We give out some of those tools. But you're right. In general, this is still too hard to do. We were just talking earlier, there's still no easy tool to use to just intersect the data in this way. All I'm saying is, I'm not really going to say it's easy to do today. But what I'm trying to convince you is that it is possible to do today. And I think if we can see the value of doing this, I think people will come and generate the tools. But I think I want more science to be done this way, to even justify these tools, besides just my lab doing it.
You mentioned when you built your database that you were using the NHGRI GWAS catalog and also read the studies. So I have
That's
two
right.
questions. Did the shortcomings or the things we didn't find in the GWAS catalog from NHGRI, did they fix those?
Yeah.
And the second question, if as you are parsing the papers, are any of the advances in how we publish papers and how we tag them better, whether they are making difference?
Yeah, it's a great question about the GWAS catalog itself. The GWAS catalog, I gave a static snapshot of it. It continues to grow and expand. In fact, we were just talking earlier today with folks from the GWAS catalog how to improve the methodology, how to learn from what we've done. And to be really clear, NIH has just embarked on an initiative now to fund extramurally a consortium to build a database that goes beyond GWAS. So, for example, I think ClinVar is a great example of the variants that at least have some clinical testing or CLIA approval or even a research-based approval, but to go even beyond that to the research tests. I think there is an initiative now, there's an active RFA, I think it's coming out imminently to really fund a larger group of extramural folks to do this. In the meantime, the GWAS catalog is great because it is a set of at least the GWAS is there, but we all have problems in how to curate this data. Now let me just make a point here too. Again, I have the highest respect for GWAS catalog. A lot of our problems come from the researchers publishing the papers. And some are quite deliberately not even telling us the SNPs that they're finding in their papers. That shows a Manhattan plot and they don't even mention the SNP. They don't mention which allele was which. And so neither GWAS catalog nor any initiative is going to easily get any data, whether it's automated or not, from those papers where investigators are deliberately trying to not show or share some of that data. I think that if I had to put any attention on anything, it's going to be that we should publish correct papers with the right genotypes and the right alleles, and we should put that data out there so other initiatives can actually start to use that data as well. So it's a long answer.
So I've got two questions. The first one relates to the CD44. And the second one is, is there going to be a diabetes cure? I'm oversimplifying. And the second one is, how do you read 100,000 papers?
Yeah, so we've already done 10,000 papers, but we'll read 100,000 soon enough. So is this going to be a cure for diabetes? Look, no one ever loves using the word cure here, right? We like therapies, and if we could get to a cure, so that would be amazing. I just think it's kind of interesting that there is something global out there across these tissues, across these species, that we've kind of missed, right? And so it's another kind of thing I've realized is when people run micro experiments, a lot of times those biologists are expecting some answer from that experiment. And then they see something that's not there that they were expecting, and they say, oh, that's just inflammatory schmutz or something that was in the sample. And sometimes that noise is the signal here, right? So I'm pretty high on this particular molecule. I think we should develop drugs against it. It turns out there's no published small molecule that hits CD44. There are, maybe something sits in a library of a pharmaceutical company somewhere. NIH is particularly good at strong-arming the pharmaceutical companies for releasing or opening some of those libraries, whether it's for repositioning or through Chris Austin's efforts, so we can get to some of those compounds and maybe screen. Is it going to be a cure? I don't know. But I like this, and we could still use some more therapies. But in the meantime, I think why I'm not a diabetes doctor, why I'm an informatician is I'm busy working on the next 10 diseases, because I think I'm going to be agnostic that way, and at least turn this over at some point to the pharmaceutical companies who want to do something with this. What was the second question, though?
Well, I mean, how did you actually physically do the 10,000 papers?
We're just going to manually eyeball them.
But
We're going to crowdsource this. You can outsource this. You can offshore this. There are people all over the world who want to read these papers for you. Okay? It's just a fact of life, okay? The younger folks know about, for example, the Amazon Mechanical Turk. We didn't Turk this. I mean, you need a higher level. These are master's level people. To be really crystal clear, I tried twice to get NIH funding to build this resource. We didn't end up getting any NIH funding on this, so I used my endowment funds to just start to build this. And I just hired a company to just start reading these. And I didn't make a big deal of it. I just said, we need this database. I'm not going to spend effort trying to figure out how to do this in an automated way. I need to get this data right away.
Is there a website yet for reading papers?
It's just manual. So how to read these papers is tricky, right? You want to know the truth? We actually curated all these 4 times, okay? We started with a high school kid, okay? For high school kid, we said 50 papers. You got all summer. You got to curate all of them. And you tell us what you think you're going to get out of this paper, because we didn't even start with a data model. We said, okay, these 50 are interesting, and we figured it's plausible to do this. So then we hired a group to just do the next 1,000. And we said, this is great, but oh man, we should have written down this, and we should have written down the ethnicity and this, so we do it again and do it again. And why am I saying this? Because sometimes when we try to set up the right perfect data model, it takes us 5 years of committees, and meanwhile, we've curated the thing 5 times, okay? So at some point, you just start doing it some way, and don't let perfection be the enemy of the good.
Yeah, here. Yeah.
I think I'm blocking you from drinks next door, so maybe we'll call it quits here.
Thank you, Atul. I'd also like to thank FAES for sponsoring this, and they're also sponsoring a reception in the library right now. So join us, and we'll have further conversation. Thank you.
Thank you. Awesome.
Great job.
Thanks.
Always is.